drug discovery is related to prevention or cure of some prevalent disease. अब वो prevalent disease क्या है, उसे कैसे remove किया जाए? We need some information जिसको हम use करें, then we strengthen that information and then we prevent or cure a disease. इसके लिए जब हम DNA की बात करते हैं, then we have a concept of SNPs, which is single nucleotide polymorphism. सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म से मुराद ये है कि किसी एक न्यूक्लियोटाइड सीक्वेंस के अंदर ऐसी प्लेस जो डिफरेंट है बिटवीन टू सीक्वेंसेस. सपोज वी हैव टू सीक्वेंसेस, अब उन सीक्वेंसेस में सिर्फ एक बेस पेयर का डिफरेंस है. ये बेस पेयर डिफरेंस क्या है और इसकी क्या एप्लीकेशन है? लेट अस स्टार्ट फ्रॉम दिस डेफिनेशन. सो व्हाट हैपेंस इज कि एक जो हमारा एसएनपी होता है, इट रिप्रेजेंट्स अ डिफरेंस इन अ सिंगल डीएनए building block, and that building block is nucleotide alone. Let us look at an example of this difference. This figure shows two nucleotide sequences. Number one is this one and the other one is the given in this block which is downwards. So now what I want to show here is we are putting the definition of single nucleotide polymorphism. अब अगर इस sequence को देखा जाए, it start from A here and T here. So which is common here also, A and then we have T. Similarly, moving on, it has A and T at these places. So this is also similar. So if I say that first strand, this one and this one, they are similar. That is right, they are similar. If I move further, then there is difference of CG here and TA here. So what is happening? That when we move forward, there is base difference of at place C, it is T. At place G, it is A. So when I am looking at these things, so we can say that in the nucleotide sequence, at some specific basis, this base changes its property. For example, C here converts to T in the second strand and G and A are convertible. So what we say that single nucleotide polymorphism basically is a change in the single base pair which is present in the nucleotide sequence. And again, this example is shown by a textual or sequence example. For example, in this particular case, we have AAGCCTA in this place and similarly this place. Now what happens is that this sequence remains similar up till here. So these first four characters are similar. Now where is the difference? Difference is in these two places and the remaining two places are same also. What is happening here is in these two sequences, we have a SNP of 1, that there is only one difference. So moving on, since DNA in a chromosome, it is a very high number of characters, we can say that in one DNA sequence, there can be many SNPs, which are shown in this part. So if I see this part, what is happening? For chromosome 1, we have this sequence, then we have this sequence, and it is going on. So when I comparing this sequence against these other four sequences, so there is a difference of this part. So this is one SNP, and similarly for these four, we have difference of this part. So we can say from this complete nucleotide sequence, we have got two SNPs which are useful or which will help us in identification or finding the difference between the given sequences. So what is usefulness? So as we know, when we want to identify difference between humans or between different organisms, we can take information with the help of this SNP. So major contribution of this SNP can be identification of humans or persons, and this is directly relevant to the domain of forensics. Similarly, mapping and genome-wide association studies of complex diseases. So अगर हमने कोई disease के effect देखने हैं, तो if they are applicable to one gene sequence, they should be applicable to other also. Estimation of predisposition of disease, कि जो disease हमारी occur होने वाली हैं, उनको foresee किया जा सके based on this gene information. अब यही जो हमारे पास SMPs हैं, they are common for identification of humans in some countries such as UK, which is shown here, and then they predict specific genetic traits also, कि कौन सी genetic traits हैं, उनकी क्या sequences हैं based on SMPs, and finally classifying patients in some clinical trials, कि यानी कि patients की information को identify करना based on some clinical trials. So now we should know some properties which are related to SNPs. Number one thing is they are present in the non-coding part of the DNA. यानी कि जो non-coding part है उसमें ये present होते हैं. Then if they are found in enough amount or their frequency is enough, यानी कि काफ़ी ज़्यादा occurrence है उनकी, then they can be useful for finding सम डिजीज, यानी कि फिर वो लिंक कर सकते हैं कि अच्छा यार ये इंफॉर्मेशन कॉमनली पाई जा रही है, तो देन इट कैन बी ट्रिगर्ड और इट कैन बी मैप टू सम डिजीज इंफॉर्मेशन। और उसी तरह फिर हमारे पास क्या है, क्योंकि इस SNPs मिलियंस ऑफ नंबर ऑफ मिलियंस में होंगे, तो इसके लिए हम क्या कर सकते हैं, जैसे हमने बायोलॉजिकल एसेज हैं, बायोलॉजिकल एसेज हमारे पास क्या है, हाई थ्रूपुट स्क्रीनिंग का एक मैकेनिज्म है जिसमें इंस्टेड ऑफ वर्किंग ऑन वन SNP वी कैन वर्क ऑन मिलियंस ऑफ SNPs इन ओनली वन गो।